Hello, everyone. Welcome. We're just going to let a few more people come in and then we will start the webinar. Okay, I am not seeing anyone else coming in from the lobby. Hi, everyone. I'm Bree Price. I'm the Extension Coordinator with the WSUB program. Thank you so much for coming to one of our webinars in our 2025 webinar series. I just have a couple announcements before today's speaker presents. The WSU Honeybees and Pollinators program is a cornerstone of the College of Agricultural, Human, and Natural Resource Sciences, abbreviated CONORS, that is dedicated to fostering resilient ecosystems in Washington and beyond. Our mission intertwines innovative research, community engagement, and education to safeguard pollinators that are pivotal to our food security and environmental health. In partnership with the Connors Resilient Washington Initiative, we're committed to advancing sustainable practices and pollinator-friendly landscapes and ensuring a flourishing future for agriculture and natural resources. We have two more upcoming webinars this year. For more information about those, please visit our upcoming events page on our website, bees.wsu.edu. There will be time today to answer any of the questions you have after the presentation. Feel free to type your questions in the box below anytime during the presentation. After today's webinar and before you close your browser, you should be prompted to answer a very short 5-question education and outreach survey. These surveys are very important for us to make sure we're tailoring our education and outreach to what you're wanting to learn about. Today's speaker is Joey Rosario. Joey is a master's student in the Hopkins Lab, and her research focuses on controlling comb storage pests like wax moth. Whether you're a hobbyist or a commercial beekeeper, storing extra comb is inevitable. How you store and reuse your comb can impact a colony's health and productivity. This webinar will explore long-term effects of comb reuse and hidden risks involved, and the common pests that target stored comb, and some efficacy of possible strategies to prevent and manage these pest infestations. Okay, Joey, I'm going to mute myself and you can go ahead and share your screen.
Okay, I will try sharing now. Here you go. Can everybody see this?
Looks good.
Okay, thank you. As Bree said, my name is Joey Rosario, and I am in my second year of my master's degree. And I'll be covering the honeycomb storage and management. And a little bit about myself. I have been part of the WSU since 2019, and I graduated last year with a general biology and fine arts degree. On my senior year, I joined the honeybee lab, and that's because I just decided that bugs are really cool and honeybees are like really, really interesting to learn about. And decided to stay in the lab as a master's. I've learned so much about honeybees, really cool facts about them, and how to manage them as a beekeeper. And with my research, I mainly focus on stored comb sanitization, and we'll talk more in depth about it at the end of the presentation. Yeah. Okay, so here's a quick overview. I'll be mainly talking about what exactly are combs and the effects of reusing combs, the main pests around stored wax combs, and what are some available management practices out there, and what my research is looking into. Okay, so first, combs are basically constructed beeswax that are made of different components like esters, fatty acids, long chain alcohols, and hydrocarbons. All of this just makes them really hydrophobic. So any water, it doesn't really stick to them. On the other hand, beeswax is very malleable, so they can easily collect debris and pesticides, which are the issues that we will be talking about later. Um, but how are they made exactly? So with after a worker eats honey and pollen, beeswax is secreted from their wax glands through their abdomen. In this photo here, you could see that honeybees have a total of 8 wax glands, and this is how they create wax scales. And they collect these scales with their tarsus and use their mandibles to mold it and create the hexagon shape of the comb. So now you could think about it like this, that honeybees are really, really small relative to us. Creating wax scales to make a whole frame of comb takes a lot of time and energy for every worker. So wax equals valuable. So, and in a comb, a cell is the most important unit because honeybees hold basically everything in there, like their food storage and any of the stages of the honeybee from like egg, larva, and pupa. And why did bees exactly choose hexagon as their comb shape? Well, there's a scholar that created a theorem called the honeybee conjecture. And so it's Marcus Terentius Varro did the math on why bees evolved to make hexagon cells. And this is because this was the most compact shape of all, and the more compact it is, meaning there's less materials bees need to use. And in this comic here, it shows that, oh, for solitary bees, they use circular shape. They're like, oh, it's strong, it's good enough. But if you need to have like a whole colony to support, you need more cells. So they try to put it where it's like a grid, but there's a lot of gaps to it. So they made it like alternating. It's like efficient, but there's still a lot of materials that's required for that. And at the very end, they're like, okay, let's make it a hexagon shape. It's like straight lines, optimized, less materials, good to go. Um, but some people ask like, oh, why can't it just be squares or triangles? But they are still considered bigger in perimeter rather than hexagon, so that's why they're not used. Going back to this, with cells, they're slightly angled upward so that the nectar doesn't spill out or the larva doesn't fall. Usually they are at a 9 to 13-degree angle. With the cell, depending on the caste, they have different sizes. So for a worker as a regular size, as you've seen everywhere, drones are a little bit bigger because of their they just have a bigger body size and have more fat bodies in them. And for queen cells, they are more likely to protrude out since they also need a lot of space. And after learning more about cell shapes, it's very interesting to see how a lot of the art designs are very, very misleading. Like in the very top photo, you could see how the tips are very much in the bottom and the top, while in drawings, you could see how the edges or the lines are in the top and bottom. And it bothers a lot of beekeepers or bee researchers a lot. So, okay. So with comb storage is very, very important and inevitable, especially during the winter when boxes or frames are not getting used. It is more cost effective to reuse comb over and over again because it takes a lot of energy for workers to make them. There is also less supplemental feeding that beekeepers have to keep up with to support the wax production. But what if it is more detrimental to the colonies to do this? And in the next few slides, I'll talk about how wax combs change over time. So first is the color change. When wax is secreted and constructed, it usually starts out as white. But over time, the wax darkens because it is absorbed whatever is put on that comb by the workers. So if there's pollen and honey, it stains the comb into a medium color, like orange or yellow, light yellow, since it does not have a lot of debris in it. But once you start putting brood in or lay eggs on the cells, this is where they tend to turn into shades of brown or black. And this is because anything from the bee development, like feces, silk, or any cocoon shells that they have, is absorbed by the wax. And this is why brood wax or brood frames turn darker quicker than supers. And it's not only that, it's also it also constricts the cell shape because of its layers. You'll see in older frames, they have a more circular shape rather than a hexagon. In the top photo here, the left side shows the full, like a newer cell. It has a more hexagon shape to it, while the right side has a more like a bullet shape at the bottom. It's more circular. And because of this, bees tend to have a smaller body, smaller morphology, which affects the majority of the bodily functions that they have. And two examples is that their legs, they usually use them to measure the how deep to make the cells and like how the diameter of it. And because their legs are smaller, the cells are smaller, so any future workers will be smaller. With this, another example is the proboscis. Because their proboscis are smaller, their foraging capabilities lowers since they cannot bring a lot of nectar into their body, their crop. And because of this, this creates a lot of work for foragers to go back and forth from the colony to the foraging area and vice versa. Okay. So this creates a domino effect where the overall colony strength is also affected. In this diagram below, it shows the different areas of the cells and how they are used. In a new or fresh comb, all of the cells are used by the workers as either as a food source for the honey and pollen cell, or as a brood cell. On the other hand, with old comb, they are more cells that are not getting used, especially in the corners. And there are a lot of pollutants that are present in the area, and this is because of like pesticides or any drift zones. And this also creates another domino effect where with less brood cells, there are less workers that emerge and less food storage. And because of this, the space is limited, and so the workers cannot fly or forage for longer periods of time, thus getting a shorter lifespan. And this is something that should be considered. Even something as small as reusing frames can even affect the overall colony's strength. Okay. So yeah, with pollutants, this does increase over time. Due to the pesticides that's applied for crops, they are affected, especially if they are at the drift zone. Okay. So next, we're gonna cover the stored combs. There are two main pests that are in Washington State. The amount and different types of pests depend on the environment. So first is the wax moth. It is an umbrella term for multiple species, but the two main ones are the greater and the lesser wax moth. So the Galeria melanella and Archaea grisella. These are very, very common pests for stored combs. If the hive is still in use, a strong colony can easily remove the wax moth and propolize the hive to lessen the invasion. But on the other hand, if it's a weaker colony, they have little to no chance to and may result in secondary diseases like black wing virus, varroa mites, nosema. They mainly damage the frame and eat the wax, pollen, honey, and brood, and leave tunnels, which I'll explain more to that with the life cycle. So first, with the eggs, they are usually deposited in the crevices after 2 weeks. And after 2 weeks, that's when the larva emerges. And this is the stage on where they are considered the most pestiferous, like as pests, because they eat a lot of the resources in the hives, such as the honey, like honey, pollen, wax, and brood. And because of this, they create a lot of whack or webbing and frass tunneling. And I'll show in the next slide. But after this, they pupate on the hive and stay pupated for 6 to 7 weeks. And once they emerge to an adult, they don't eat any of the resources in the hive. Their main goal is to mate and lay the eggs and repeat the cycle again. But here are the main damages that they have. On the very left side, it shows like the pupa, how it creates dents into the box, which is not ideal in hives because it creates more spaces for the bees that they need to use their propolis, which is kind of an important resource that they have. The middle top photo shows a group of larvae in the very corner eating all of the resources that as much as they can. And you can see some slight tunneling around it, but they usually like to clump and just eat whatever is in there. They're just like it looks like they're indestructible. They could just whatever's on their path, they just go for it. The middle bottom photo shows like the pupil casing and how this is how they usually are formed. They're like in rows, and that's how they create the dents. And finally, on the right is the more prominent tunneling. You could see the white webbing. That is the silk that they produce around it. And the red, you can see a little bit of the yellow. It looks like pollen, but those are the frass. And usually, the best way to tell that you have a wax moth infestation is through the white webbings rather than the frass, because of how similar they look to the pollen. Okay, so next we're gonna cover the small hive beetles. These are recently emerging pests in Washington state, which was surprising since their ideal environment is usually in the higher temperature and humidity. So like the southeastern states like Florida and I think North Carolina has them. Um, these are the only species in the Nitidulidae family or like the sap beetles that relies on honeybee hives. So if I put them anywhere, I don't know, like somewhere that has flowers, they won't even care about it. They just as long as it has wax or like a hive, that's where they thrive. Similar to the wax moth, the small hive beetles are destructive during their larval stage, where they eat everything in the hive except for the wax. And they spread the yeast all over the hive and spoil the honey and attract other small hive beetles. But if you would like to know more and read more about small hive beetles and how to manage them, a team in our lab wrote this extension publication. You could scan it in this like corner, the QR code that will be available in this slide and a few more slides. So, and let me know if the QR code is too small, and I can enlarge it. But in that publication, it shows like a heat map of where the small hive beetles are more likely to thrive in Washington state. The red and orange areas are the more suitable air habitats, while the blue and purple are the least. So here, the higher habitat suitability are at the lower elevations near the Puget Sound, so it's more on the west side, while the central and eastern areas of Washington state are less suitable because of its arid and desert climate. It's too dry, so they cannot survive through that. Here is a diagram of the small hive beetle life cycle created by Bree. It shows here first is that the female small hive beetles lay clusters of eggs in the crevices of the box. And once they hatch, the larva just eats anything that's on site and releases this yeast that makes the honey pungent and makes them fermented and very slimy. So we can't really use this honey once it's at this state. It just looks really gross. Um, And once they are close to pupation, or once they're close to cocooning, they're at the wandering larva stage, and they just wander around the hive until they find a place to burrow and pupate, which is usually outside the hive and under 4 inches under the soil, underground. During the pupal stage, this is like the most vulnerable and longest part of their cycle, usually spanning 75% of their life. And when they emerge as an adult, they go back to the same host hive and repeat the cycle. And again, with the yeast, because of the sliminess and how fermented smell of it, that's how the adult knows that they can go back to this hive and there are resources available for them. But if there is an active colony in the hive, honeybees typically can remove them, but because of how small they are and if there's a lot of crevices in the in the box, that's where they can easily hide in the crevices. Okay. And other pests that we have are the ants. They're not really that detrimental to the wax or like the materials, but they're mostly considered as a nuisance to beekeepers. And the next one is our mice. Usually they create a nest inside the hive and use it as a shelter. And this could be more dangerous to beekeepers, especially with the presence of fecal dropping. So if you're cleaning out your stored combs and all of a sudden you see frames with like these random, what do you call it, clusters of nest, and you just get that poof, you're more likely, you have to be really careful when it comes to that. And this is where personal protective equipment is very, very important. Okay. So next, I'll cover the management strategies that are available out there and what are possibly we're looking at. Okay. So one big factor that I've been showing you guys, or like putting out there, is that it is beneficial for future colonies to only reuse the frames for 3 years. After that, it will be best to throw away the frames to increase the colony strength, and they don't have to work with frames that have debris or pesticides embedded in it. The main downside of this strategy is that it'll be very, very expensive. A frame or like, and it'll be also expensive for the workers to remake their wax again. But this is also like a something to consider just for their like money versus like the strength of your colony. You can weigh that out. But another possibility is you could scrape off the old combs on the frame, but that's not very ideal in a commercial setting. So. Another possibility is the cold storage, which is a good cultural control for mainly for wax moth. According to this paper from Zhu et al., the table shows the different ages of the eggs, like 1 day old, 3 days, 5 days, 7 days old, um, and their mortality percentage at a specific time, whether it's like 1 hour, 3, 5, 7, 10 hours. And also compares the negative 15 degrees Celsius versus negative 20, and seeing whether one is better than the other. But what this paper is basically saying that freezing equipments in either negative 15 or negative 20 in degrees Celsius for 10 hours would kill the wax moth in all stages. It just shows that there's 100% mortality in all stages or in all days. But when it comes to larva, pupa, and adult wax moth, you can easily kill them in like an hour in either one. But eggs are like the most frost resistant, and they're the ones that need more time. There are two ways to handle this. You can either freeze all your equipment in like 10 hours, or you can even leave it overnight, like for 24 hours, and all the stages will be dead. Or you could just freeze all the equipments in an hour, and then after 1 week, you could freeze it again in an hour so that all the eggs that emerge into larva like that will also kill them instantly. Okay. So next is the another popular approach is the biological control, and this is the Bacillus thuringiensis, or BT in short. It is well-known agricultural biopesticide because it has a protein that is highly toxic to some insects, like butterfly and moth larvae. So with moth larvae, that's really, really good for wax moth. It is naturally occurring in soil, so it won't become invasive in the future. And their mode of action is having these proteins bind into the receptors of the insect, which causes the cell function to disrupt and kills the insect. It just makes their bodily function like really wonky and just kills them through that. And the best part about this option is that it doesn't affect the pollinators, making it really a safe alternative uh, um, compared to chemical pesticides. So this is what is really well known. There is like certain or B402 that's being used. So this one's the pesticide use, the chemical control, or using synthetic pesticides, which are used as a way to combat really stubborn pests. When it comes to using pesticides, it is important that you read the label and follow the instructions, especially wearing the proper personal protective equipment, PPE in short. And this is because a lot of it can go wrong with using pesticides. They can build a lot of resistance and it could be damaging to your health and also the pollinators' health. So it's very important. Um, with the bee health and was it usually it's bad for bees, or majority of this pesticides are pretty bad for the bee health in general, and it should only be used once the frames are in storage. So for wax moth, there's the paramoth, which uses the PDB. It is a fumigant that is usually left in storage supers. It cannot be used with live bees at all. Um, Same with the small hive beetles. There's the Checkmate Plus and the Whitex. These two are explained further in the publication extension, but they are approved in Washington State, but they are highly toxic to bees and are only used as a last resort if the small hive beetles took over the hive. So beware. So this part is my research. It's like an overall showing like what I'm working on. And as you can see here that at the very left, it shows like the bacteria, the larvae, viral diseases, and varroa mites going into the honeybees, the colonies, which once they're affected and the colonies pass away, like you think it's the end, but actually the hives are still contaminated with these things. The main thing I look at is the American and European foul brood, which are highly contagious. So one of the three options of how you can manage them is first is burning, but this costs a lot of money over time since once you burn the equipment, you can't really, sometimes it's harder to use them or it just can't be used. Second is putting antibiotics, but over time that can cause antibiotic resistance. So it's not really a good constant repairment. And third is just like overall like chemical or pesticide use, but that can create residues, which could be bad for us as consumers of honey. So the three options that I'm looking at here are ozone, peracetic acid, and chlorine dioxide. With ozone, all three of them are fumigants, so they're just applied through a big chamber, and I just put all the materials and see whether it removes American European foul brood. And I'm also looking at wax moth and small hive beetles in this case. But for ozone, it's pretty well known to use as a sanitizing agent for other fields. So I think it's used for onions, tomatoes, or potatoes. And same with the peracetic acid, that's used mainly as a wash. So seeing how this is used as a fumigant will be interesting to see in for wax and like any stored equipment. because parasitic acid's been used in the medical field, mainly in dentistry, as a sanitizing agent for their tools. And for chlorine dioxide, that's mainly used as a surface cleaner. So it'll be fun to see how that gets infected, how that changes like the surface of hive boxes and frames. But I think for wax, it might be more difficult since that's where a lot of the pesticide or diseases are mainly embedded within inside the wax. So I don't know if that will be as useful as a pesticide or as an option. But that's all I have. Thank you for listening. Do you have any other questions?
It looks like we have one question about your slides about the cell shape. So as the cells change shape over time, does the queen bypass those cells to lay an egg? Like, does she show a preference?
No, she doesn't show a preference because if the cell is that sh like all of the cells in that frame is in that shape. If she doesn't have a choice that but to lay an egg there, she wouldn't care as much about it. Like there's no preference to it. So
Does using a home freezer work for freezing the frames?
as long as the freezer can go to -15 or -20, that should be ideal because I think a lot of freezers can, if it's like a big freezer, like a when you open inside, you should be fine. But that's where you just have to look out on like what temperatures it can go for is the main thing with when it comes to freezing um the hives or the equipment.
So maybe put a thermometer inside
Yeah,
or something.
yeah.
Okay. What is your preferred method to replace the wax in frames?
Um, to replace the wax. Another way is usually if you want to save the frame, the wooden frame around it, you could just another way is like you could kick it and remove the middle and then replace it with a foundation, and then that's where the workers can rebuild their own um, cells again because or you could scrape it. So there's either scraping and then leaving the foundation in there, or you could kick out the foundation and the rest of the wax combs and then just put a new foundation in. Those are the two best options.
What is the best way to store your supers over winter or when they're not in use?
I think with that you could just leave because usually what we do we just leave them with the along with our other regular boxes. Like we don't really do anything special when it comes to keeping supers unless you have. But yeah, as long as they're indoor and they're stacked and you just keep track of the pest by applying certain or BT before storing or putting it in the freezer before putting them in the final destination.
Is there a best method or a best practice for storing frames?
Usually, just, as long as it's indoor and a lot of it's preventative. So as long as you make sure that um maybe at the big before storing them in a container, you apply the you can apply BT, or you can even put the paramoth um inside throughout the season or throughout the winter, and then midway through check it again, and then before you start check it again. It's a lot of checking in between.
How often do you change frames and foundations? Every year?
In our case, because it's also hard to get the materials, unfortunately, we do not really change it as often. That's where this is like a ideal situation where if you change the frames often, the colonies would get better. But in a actual setting, as of right now, it's harder to achieve that because materials are expensive right now, and um, with our colonies, they also are more reliant on ones that are already used that we have, unfortunately.
I know one recommendation our apiary manager had was replacing about 30% of your comb each year. So you don't always have to do all of it every year. But that was
Yeah.
the standard that he recommended.
Okay, yeah, that's a good idea too. Just so even like slowly changing it over time is good. It doesn't have to be all of a sudden like all materials gone, because that like your colony would not thrive at that point.
Are there any other questions? Okay, if anyone thinks of any other questions after the webinar, you can email Joey. Joey, do you mind putting your email in the chat box? I believe it's just joey.rosario, right? At
Yeah, it's just
wsu.edu.
I'll put it in there. Yeah, it's just Gina.
Oh, okay. Yeah, j.rosario at wsu.edu.
Mm-hmm.
Okay, thank you so much for attending this webinar. Remember, before you close your browser, you'll be prompted for that short 5-question education and outreach survey. And as another reminder, we have 2 more webinars planned this year. Our next one is in September, and then the last one's in October. For more information about those, you can go to our upcoming events page on our website. And if you want to rewatch this or tell your friends about it, all these webinars in the series will be recorded and posted on our YouTube channel. So I'll be uploading that within the next couple of hours. So thank you so much for attending.
Thank you.